Hi, everyone. Good afternoon. We're going to get started right now on lecture number 8, which is heredity. So we're almost nearing the end of the semester. We only have 2 more lectures to go after this one. There's number 9, which is on biotechnology, and then we will have the bodily tissues, which is a nice transition to the next semester. So in this study guide, we have some pictures that shows the different stages of meiosis. So just to refresh your memory, meiosis is a process that is used to produce gametes. Gametes are these sex cells, right? So sperm or eggs, and that takes place in the reproductive organs. Okay? So we start with a germ cell here, germ cell, which is either a spermatocyte or oocyte in female, right? Okay. So these are diploid cells with 2 sets of chromosomes. We can draw some of these things out. So 1 set of chromosome, and then we have a second set of chromosome. And of course, in a actual germ cells, we would have 46 of these things, right? But for simplicity's sake, we are just gonna keep it simple. Okay, so chromosome 1, the red one from mom, the blue one from dad, and then chromosome 2, right? And then what happens is we will duplicate the genetic material. So afterwards, we will have the sister chromatids stuck together, okay, like so. Okay. And you will separate them. You will separate them in meiosis number 1. Meiosis 1 is the first division, and afterwards we would get 2 cells. Okay, now there might be some exchange of genetic material that would be going on here. You know, some of the red will get to the blue one, and some of the blue will get on the red, right? This is called crossing over, which creates genetic variation, right? So afterwards, when you separate the chromosome, you might end up having a pair like this, right, with a bit of blue stuck on it, and then you will get the small one as well, but this one has a little bit of red stuck in it. Right? Similarly here, you would have the blue one with a bit of red stuck on it, right? That just represent exchange of genetic material between the 2 chromosomes, right? So there you go. This is the product of the first meiotic division. And then what's going to happen is these will go on and divide one more time, and you will end up having 4 cells. Okay, this is meiosis 2. And in this case, the sister chromatids will separate. So one of them might get that. Okay. And then here we are going to have the short one as well. Long red. Excuse me. And we will have this short one like that. Right? Similarly, if we separate the chromosome here, we will have a cell that has the blue one, and then the other one might be the blue one with a little bit of red. Right? So all these are random processes, and we end up having 4 separate cells. Okay, so this is a germ cell, which is 2N or diploid, which means there are 2 sets of chromosome, and then Over here, they are going to be haploid, which is just one set of chromosome. And of course, these are genetically different, right? And if we're talking about male here, they would be sperms. And if it's female, then you would get one egg. The other three are going to be polar bodies, okay? Which I can explain again later on. But that's pretty much like the overview of meiosis, right? Now let's take a look at these pictures and see if we can identify some of them. So the first one here, we can actually see there are 4 cells. That means you have gone through 2 separate divisions already. And so this is going to be telophase. Telophase number 2. Okay. Here we can see 2 cells, 1 cell, 2 cells, and the chromosomes are being pulled to opposite ends. Right? So the chromosome gets separated to opposite ends during the anaphase, right? But because we see 2 separate cells doing the same thing, so we know it was after the first division. That's how we know it's anaphase number 2. Contrast to this, this is only a single cell, and we have the chromosomes moving to the opposite end. That would be anaphase 1. Okay. And the next one, we're gonna have just the chromosome lying around in the center, not doing much. That would be prophase. And we only see 1 cell. That's prophase 1. Over here, 2 cells. 1 cell here, 2 cell here, and that would be the telophase 1. Right? That's just when the cell have finished doing their cytokinesis. 2 cells separately. All the chromosomes are lined up in the middle, so we know that is the metaphase. 2. 1 cell, all lined up in the middle. Metaphase 1. And here, this is just after telophase 1, and then the chromosomes all go back into the nucleus for a moment there. That would be the prophase 2. Does anybody have any question? Okay. So over here, we have a table, and it tells you the number of chromosomes before meiosis. So the number of chromosome, is it diploid or is it haploid? Well, you should remember only diploid, only cells with even sets of chromosome can undergo meiosis. Right? So like if something has 3 sets of chromosome, that is not gonna undergo meiosis because you can't divide 3 by 2, right? You cannot have like 1.5 set for each cell. That's not gonna work, right? So you need to have even set of chromosome. So in this case, it has to be diploid. Okay, diploid. So 46 chromosomes, number of homologous, number of pairs of homologous chromosome, that would be 23. Okay, 23 from mom, 23 from dad. That makes up 46. After meiosis 1, how many chromosomes do we have? Now, this is a little bit of a trick question, okay? This is going to be 23 chromosomes, and it's going to be haploid. Now, why is that the case? Let's go back here and see. Okay? Now you understand the beginning is 2n, and then the n is 1n, right? Because there are 4 chromosomes here, 2 sets, right? So if I put the numbers like chromosome 1, chromosome 1, chromosome 2, chromosome 2. So you have 2 of each. That's why it's called 2n. Over here at the end, you only have 1 of each, 1 and 2, 1 and 2, 1 and 2, 1 and 2, right? So you only have 1 of each chromosome number. You have 1 set. That's why it's n. But what about after meiosis 1, right? Let's take a look here. How many chromosomes do you see in this cell? How many chromosomes here? Can someone please tell me?
2.
That's right, 2 chromosomes, exactly, right? So there's 1 chromosome here, and then another chromosome here, right? So this whole thing, because they're stuck together, we only count it as once. Count it as 1, right? So that's chromosome 1. It's not 1, 1, it's 1, because they're stuck together, and then chromosome 2. So here, we just have 1 set, right? So this is still considered as haploid. Okay, it's 1n. So we started with 4 chromosomes, and then this is going to be like 2 chromosomes. Right? And after they split, it is still 2 chromosomes. All right. So over here, after meiosis 1 is 23, and it's already haploid because you only have 1 set. And then after meiosis 2, it is still going to be haploid, and it's 23 again, right? But there are no more homologous chromosomes, right? You no longer have a pair. And that's the whole point. You're trying to reduce the number of genetic material by half so that, you have 23 in sperm and 23 in eggs. When they come together, 23 gives you back your 46, right? I'm just going to do one more example here. If you have 18, the earthworm has 18 after meiosis 1. That means originally it would have 18 pairs of homologous chromosome. And then it would be 36 chromosome in the diploid cell. And then 18 and 0. Okay, so basically the first number, if you divide it by 2, then you get all the other ones, right? Okay, I hope that makes sense. Anybody have any questions? All right, moving on. Complete the following chart to compare mitosis and meiosis in humans. So first one, the number of division, right? So for mitosis, there is only 1 division. 1 division. Meiosis, on the other hand, have 2 divisions. Okay, meiosis 1 and meiosis 2. Number of daughter cells produced, it will be 2 genetically identical daughter cells. Okay, they are exact clones of each other. Whereas for meiosis, it will be 4 genetically different cells. Okay, because of the crossing over that happened during prophase 1. Now, the daughter cells, are they diploid or haploid? Well, for mitosis, we are getting the exact same cell as the original, so those will remain as diploid. 2 sets of chromosome, right? 2n. And for the meiosis, we are going to have haploid cells. 1 set. Number of chromosomes in daughter cells, 46. It's the same as the starting one, right? And for the meiosis, it will be 23. So what's going on during prophase? Okay, so in prophase, the homologous chromosome, they don't find each other. Okay. Like chromosome 1 and chromosome 1 are just like on their own. They don't come together, right? Chromosome 2 and chromosome 2, they are not together, right? They're replicated, they're cloned, but they're not gonna, hang around next to each other. Okay, so homologous chromosomes do not find each other. They are just randomly scattered in the cell. On the other hand, in meiosis, the homologous chromosome means like chromosome 1 and chromosome 1, right? Chromosome 3 and chromosome 3. Chromosome 7 and chromosome 7. They will find each other and form these tetrads. Okay. So homologous chromosome form tetrad. And once they form the tetrad, crossing over occurs to create genetic variation. Okay, so we don't all look the same as our parents. All right. So next, we have the metaphase. Okay, so in mitosis, the chromosomes will line up in the middle. Okay, so like you might have a X here, and then like the smaller one represent chromosome 2, right? So they all line up in the middle. Okay, so chromosome line up in the middle of the cell, of course. And that would be the centrioles here, and then you will have the spindle fibers, right? Like that. For the meiosis, we will have the tetrads line up in the middle. Okay, so it would look something like this. Right? We would have the X, the maternal one, and then the paternal one, like that. Right? The tetrad lines up in the middle. And then the next one, same thing, right? Now, take a look at this. It's random how they're gonna line up. Just because the red one is on top here doesn't necessarily mean the red one's gonna be on top for the next pair. I mean, it could be, but there's no rule for that. Okay, so like it could be the blue one is on top, right? It could be the red one is on top. So each how each tetrad lines up is an independent event. Okay, meaning it's not affected by how another pair lines up. It's like flipping a coin, right? Like whether you get heads or tails on the next flip has nothing to do with whether you got heads or tail on the current flip, right? They're independent events. Okay, so the tetrad lines up like that. We're gonna have our centrioles, and the spindle fibers. Okay, so those are the major difference that I want you to be aware of. Okay, genetic relationship of daughter cells to parents, identical. And this one is different because of the gene shuffling that occurs, as well as the random alignment of the tetrads, right? Type of cells involved, these would be somatic cells, bodily cells. These are germ cells. Okay, so things that will become the sperm and the egg, those are called germ cells. Function, purpose of mitosis is for growth and development and repairing, right? Repairing damaged cells, and asexual reproduction. On the other hand, meiosis is for gamete production, right, which is needed for sexual reproduction. Asexual means there's only one organism needed, right? They basically divide and clone themselves. Sexual means you need to have two individuals, okay? Any question? Okay, that's that. So let's take a look at law of independent assortment. So there is this person named Mendel, right? The so-called father of genetics. He's the guy who came up with a lot of these, discover a lot of these, patterns with inheritance. Okay, and he has two laws. The first law is called the law of independent assortment, which is what I was trying to tell you, right? Like whether the tetrad lines up as like red on top or blue at the bottom or the other way around, those are not related to each other. Okay, so basically, on those chromosomes, you have a bunch of genes, right? These genes code for proteins, of course, and so the law of segregation, sorry, the law of independent assortment basically states that, let me choose the right color. It states that the sorting of genes into gametes are independent events. Okay. For example, let's say we look at two genes. Okay, consider big A, small a, and big B, small b. Okay? So we're gonna produce some gametes here. Okay? We're gonna have some gametes here. All right. So what are some combinations here? Right. Well, we can have big A, big B, right, for one of the sperms, one of the gametes. It's also possible to inherit a big A and a small b. Right? What are two other combinations here? Can someone help me, please? What other possible combinations if we're just looking at two sets of genes here? That's okay. You can have small a, big B, or small a, small b, right? So whether you get the big A or not has no effect on whether you will get the big B. Just because you inherited the big A doesn't necessarily mean you're going to get the big B, right? You can have a big A and a small b, right? So how one gene A, sorts into the gamete has nothing to do with how gene B sorts into the gamete. We call this the law of independent assortment, okay? He has the second law, which is the law of segregation. Okay? This one is pretty straightforward. It states that diploid cells contain pair of alleles. Alleles are different versions of a gene. And these alleles separate during meiosis, right? And it gets passed down. Right? There are two A's, your big A, small A. You either get the big one or the small one, not both. Okay, it separates. So that's the law of segregation. Okay. Does anyone want to ask anything before we move on? Just gonna take a look at whether or not you guys watch the video recording. I want to see if you guys just not getting this or.
Vincent, I didn't see the video you posted on Monday. Did you post a video on Monday?
It's all online already. I don't have to put anything, right? Everything for the course
Oh.
is already online, right? Okay, so like all you have to do is go to the lecture folder and everything you need is already there, right? Okay. Anyway, we're gonna have to continue over here. Maybe I will just do a quick poll. Okay, can you let me know if you have watched the video or not? If you watched it, please choose A. If you have not, please choose B. It's anonymous. I wouldn't be able to tell who's who. Let me see. Okay, so only a few of you have done that. I mean, that's okay. You can just see if you're able to follow along. And you know, as usual, you should watch the recordings to get the full picture of what's going on. Okay. Give me a second here. Okay. So this next question has to do with whether the inheritance pattern is dominant or recessive. Okay, so if you think about two allele, right? Big A and small a. The big A is the dominant allele, meaning it will always show up, whereas the small a is a recessive allele. You need to have two copies in order for the trait to show up. Okay, so here is a pedigree, and we want to see whether the inheritance pattern is dominant or recessive. So there are a couple of like key things you can notice to see whether someone is dominant, if a trait is dominant or recessive. Okay, so for example, over here for the first example here, this individual has the trait. Okay, and her parents does not have the trait. Okay, so that is a key pieces of information that tells you it is a recessive inheritance pattern. Okay, so this person is small a, and then the parents is big A, small a. This is the only way for it to occur. Okay, so if an individual expresses a trait and the parents do not, then it is recessive for sure. Okay. How do we know it is not dominant? Okay. So let's for a second think about what dominant would look like. So if this trait is dominant, that means there must be at least one capital A, right? It could be big A, or big A, small a. We're not sure. But the fact that there is a big A means at least one of the parents would also have a big A. And if it's a dominant condition, then at least one of the parents would also have the trait, right? Okay. So if it's dominant, you cannot have a child who has the trait, but the parents don't have it. Okay? Like if the kid has it and it's a dominant trait, then at least one of the parents must have it. Okay? So let's take a look at the second one here. Like, is this dominant or is this recessive? You know, you can test it out first. You can guess. Okay, so let's say we guess that it is recessive. Just a wild guess. Right? So if it's recessive, the only way for you to have the condition is to be homozygous, right? Little a. So if you have little a here and little a here, then that means all their kids must also have the condition, right? But they don't, right? So this tells you it cannot be recessive. It's not possible. If it's recessive and both parents have it, then all the kids must have it. So that tells you this must be a dominant trait. And because the kids don't have it, the parents must be heterozygous. And these kids are going to be small a, like that. So this is dominant. Right? In case I wasn't being clear, this is recessive. So if both parents have the condition, have the trait, and at least one of their kids don't have it, it is dominant. Okay, what about this last one? Based on what I just taught you, do you think this is dominant or do you think this is recessive? What do you guys think? Take a look at this last one right here. Well, you can see that, again, you have to look at the parents, right? And look at the parents and look at the kids. And you can see that over here, we have parents who don't have the trait, but then their kids have it, right? So according to what I told you, if an individual expresses a trait and the parents don't have it, then it must be recessive. Okay, so this is little a, big A, small a, like that. All right, so that's the solution for the pedigree. A pedigree is a family tree, right? Okay. So now we're going to move on to solve some genetics problem. And I think I'm just going to choose a few here, and then we'll take them up. All right. So let's say we have this one.
Excuse me, Vincent. I have a question.
Sure.
Regarding the first one, the recession, right? Is that
Recession?
in another word? Yeah, in another word, that means the parents don't have any symptoms like a trait, right?
That's right.
But
they don't
they
show
are
it. Exactly.
Yeah, doesn't show. Yeah, but they are carriers.
Yeah, carriers.
Is
Exactly. That's right.
Okay?
So
Okay.
if you have an allele, you're carrying a allele, but you don't show it, then you are considered as a carrier of that allele. Very good.
Okay.
Yeah. So let's take a look at this problem here. We'll learn how to solve that. In peat plants, tall is dominant over short. Okay, so you want to write these out. You want to be like, big T is tall and small t is short. Okay, big T is tall, small t is short. What is the expected percentage of offspring for a cross between hybrid tall peat plants and pure tall peat plants? Okay, someone asked me to explain dominant and recessive again. Sure. So allele is different versions of a gene. Eye color, for example, there are brown eyes, and then we have blue eyes. Okay, so these, and then there are other eye color. Okay, but let's just stick with blue, brown, and blue. Right? Now we are diploid organisms, which means you have two sets of chromosomes, and so on the eye color gene, you might inherit big B, big B. Right, so this one is easy, right? Big B, big B. What color would the eyes be if someone is big B, big B? Francisca, since you asked this question, can you answer it? What's the eye color for someone with big B, big B? Are you there, Francisca? My question is, if you get big B, what's the eye color? Is it brown eye or is it blue eye? It says right here, right? Brown eye, that's right. Okay. So if the alleles are the same, we call it homozygous, okay, or pure. What about small b, small b? What's the eye color? Well, small b is blue eye. Also homozygous, right? So the confusion occurs when you have big B and small b. Okay, so these are homozygous. And if it's different, then we say heterozygous. Heterozygous. So what color would it be? Since brown eye is dominant over blue eye, then that's the one that's going to show up. Okay, so dominant means it's the trait that always shows up. Okay, it doesn't matter if you just have one copy or both copies, it always shows up. For the recessive one, you need to have both copies present in order for it to show up. Does anyone have any follow-up question? Okay. So here we have the hybrid tall plant. Hybrid means heterozygous. Okay, heterozygous means different, right? So we have a big T, small t. That would be a hybrid tall plant. And then we will cross it with a pure tall plant. Pure means homozygous, same. So that's big T, big T. Okay, so when we do these kind of questions, we want to create a Punnett square. Okay. So these alleles will separate during meiosis. So one gamete will get big T, the other gets small t. Same thing over here, big T and big T. So if this and this comes together, if the gamete and this gamete fertilizes each other, then you will have big T, big T. This and this can come together and also get big T, big T. Same thing here, big T, small t, big T, small t. So what does it mean? Well, 2 of the 4 boxes is going to be big T, big T. So that's 50%. 50% of the offspring will be big T, and that's going to be a tall plant. The other 50% is going to be big T, small t, which means they are going to be short plant. Okay. So the genetic makeup is called the genotype, and then the appearance is called a phenotype. Okay. So 50% a big T, small t, and then 50% big T, big T. Oh, sorry, these are also tall plants. My bad, I misread it because big T is dominant, right? Okay, so 50% big T, 50% big T, small t. But you can see all of them are going to be tall plants, so 100% tall. Okay. So the genotype is the genetic makeup, whereas the phenotype is going to be the manifestation, the expression of the gene. Let's try a second question here. Black coat color in guinea pigs is dominant over white coat color. Show the results of the phenotype and genotype for the following cross. Okay. So again, we have to set up our letters. Okay. Because black is dominant over white, we put big B for black and small b for white. We have to use the same alphabet. Okay, don't use B for black and W for white. It doesn't work like that. Okay, we have to follow the same alphabet as the dominant gene. So here we have a hybrid black pig, and a white pig. Okay, so what's a hybrid black pig? Can someone tell me, please? What's the genetic combination for hybrid black pig? That's right, Sarah. It is going to be big B, small b. Thank you. We are going to cross it with a white pig. Because white is recessive, they don't have to tell you whether it's pure or hybrid, right? There's only one way to be recessive, right? That is to be small b, small b. So we're going to go ahead and do the Punnett square. Okay, we basically take this one and we separate it. We have a big B and a small b like that. That means half the, let's say it's the male guinea pig, right? So half the sperm will carry the big B, the other half will carry the small b. And for the female pig here, half the eggs will be small b, the other will be the same thing because that's all she has. So we're gonna combine these. If this sperm fertilizes that egg, we get big B, small b. Same thing here, big B, small b. Small b, small b. Small b, small b. So let's take a look at the genotype. The genotype will be 50% big B, small b, and 50% small b, small b. What color would they be? That would be the phenotype. So if it's a big B, then it's going to be black because big B is dominant, right? As long as you have a big B, it's going to come out as a black guinea pig. So that's 50% black and 50% white. Okay. Let's do one more here. Let's do the C here. Hybrid pig is mated with an hybrid pig. Hybrid means different, right? Heterozygous. So we are taking big B, small b, crossing it with another big B, small b. Whenever you have a hybrid pig, it's gonna be black. It's the big B that's gonna show up, right? So once again, we're gonna do the Punnett square. Okay. This pig will have two kinds of gametes. Half of it will carry the big B allele, the other will carry the small b. Similarly, big B and the small b. So we have big B here, big B, small b, and then big B, small b again, and then small b, small b. Let's take a look at our genotype. 1 out of 4 boxes is big B. That's 25%. 2 out of 4 boxes is big B, small b. That would be 50%. And lastly, we have small b for 1 of the boxes. So that is also 25%. What percentage would be black? Please help me out here. What percentage of the guinea pigs will have a black coat?
25%.
And how did you get that?
Because there's big BB, right? The one big BB there.
Yeah, that's the, yeah. You want to change your answer now?
Oh.
No, that's okay. You can change it. Or anyone else? What percent would be black coat? Okay, so 25% will be black here, but these are also black, right? Because as long as there is a big B, it's going to be black because the big B is dominant. So 25%, 50%, that's 75%.
Oh, 75.
Yeah, that's okay.
75, yeah.
Yeah, 75% black and 25% white. Okay, so the phenotype is not necessarily the same as the genotype. Okay. And if you're not sure about these terms, if they sound unfamiliar, it's probably because you didn't finish watching the recordings, right? Okay, so I said many times, the tutorial that we're having right now is not a substitute for the lecture, right? Okay, so let's move on and do another example here. In humans, brown hair is dominant over blonde hair. If a woman with brown hair whose father has blonde hair marries a man with blonde hair, what is the probability of having a child with brown hair? So it's a little bit complicated. Maybe we can use a pedigree to represent the situation here. So square is man, circle is woman, and that's to get married. Okay. So we have brown hair, big B, and then small b is blonde hair. So we have a woman with brown hair. Now, if you just say someone has brown hair, we don't know whether it's big B or big B, small b. Okay, but we know for sure there is at least one big B. So big B something. But then the question says, her father has blonde hair. So if we go back one generation, right? So this would be like her father and her mother. We don't know anything about her mother, but we know her father has blonde hair. And there is only one way to get blonde hair, right? Which is small b, like that. Okay. So what does that tell you about the woman? Well, if her father has small b, that means he can only pass on the small b to his daughter, right? So it's guaranteed she will get one small b from her dad. Right? So based on this information about her dad, we know she is going to be heterozygous. So who does she marry? Marries a man with blonde hair. Blonde hair, there's only one possibility. It must be a small b. So now they're gonna have kids. I want to know what's the probability that the kid will have brown hair or blonde hair. So we do the Punnett square again. For the man, the husband, he's blonde, so he can only pass on little b. All of his sperm is gonna carry the little b allele. And for the woman, the mother, she has big B and small b, so half of her eggs is going to have big B, the other half is going to have small b. If this sperm fertilizes that egg, we're going to have big B, small b. Same situation here. And then small b, small b, small b, small b. So it's going to be 50% big B, small b, 50% small b, small b. Okay, so in this case, this is going to be brown hair. This is going to be blonde hair, right? 50%, 50%. Does anybody have any question about any of the stuff so far before we move on to the next part? It is going to be more complicated, the next part. So please, please, please ask if you are a little bit confused. All right, no confusion. That is good, I suppose. So let's go back and try a different question now. We have a cattle with long horns. So let's copy this. Actually, you know what? Before we do that, since a lot of you didn't watch the video, it's important we go through this thing. Okay, so like a dihybrid cross. Dihybrid cross means we are tracking two genes at the same time, right? Two separate genes. So instead of just like, big B, small b, we're tracking another one, maybe like, big A, small a or something like that. So let's say we have some plants, okay? Tall is dominant over short. And then we will say red, as in like the flower color, is dominant over white. Okay. So let's say we have two hybrid plants. Okay, two hybrid plants. So what would the hybrid plant genotype be? Hybrid means they are different, right? The two alleles. So it would be like big T, small t, big R, small r. So that's going to be a tall red plant, but hybrid because it's like different alleles, right? For the T and different alleles for the R. So let's say we have two of such plants. We're going to cross it with another big T, big R, small r, and other tall red plant. Okay. So we want to know what is going on here. Now you want to copy this down. This is a very important chart that we're going to make. right? So let's see what kind of gametes we will be able to produce. Okay. So we can have a big T and a big R. That's one possible gamete. We can have a big T and a small r. We can have a small t and a big R, a small t and a small r. So those are the 4 possible combinations. Similarly here, we will have the exact same thing because they are the same genetic makeup, right? So big T, big R, big T, small r. It's kind of like doing those polynomials in your math class, right? Okay, so now we're gonna make a Punnett square, but this Punnett square is going to have 16 blocks, right? Let me see if I can do it here. Insert table. I'm gonna go 4 by 4 like that. Okay, so let's see if it's if I can make this bigger. No, it doesn't really work. I will just have to draw it myself. Okay. So let's put the gametes on the top here. Big T, big R. Okay. And the next one is going to be big T, small r. And we will have small t, big R, small t, small r. And on the side here, we would have the same thing, but from the other parent, right? So if this fertilizes with that, we're gonna have big T, big R, big R. You take this one and this one, combine it, we will have big T, big R, small r. We keep on repeating this process, and we will see all the various combinations. So again, over here, big T, small r combined with big T, big R. Right? Big T, small t, big R, small r. Big T, small t, small r, small r. Okay. Let's try the next row. Small t, big R, small r. So in the end, we would have 16 different genotype. Like that. Right? So it's too difficult to map out the different genotypes here, but we can figure out the phenotypes. Okay, the phenotypes. Okay. So let's highlight this one. What is this plant gonna look like? Is it tall? Is it short? Is it red? Is it white? What's it gonna be? Someone tell me. What would this be?
Is tall and red.
That's right. Big T is tall, right? And big R is red, right? So this is tall and red. Now we're gonna go and try to find everything that's tall and red. Next one here. As long as you have a big T is tall, as long as you have big R is red. So that's tall and red, tall and red, tall and red. All these are tall and red, as are these. Okay, there is another tall and red. There is another tall and red. So that's 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 out of 16. 9 out of 16 are tall and red. Okay, those are the yellow ones. Okay. What is another genotype that we can, another phenotype, sorry, another physical appearance that we can look for? Anyone else want to try? Like, what would this one be? What does it look like, the plant?
I think it's also be tall as well.
Tall as well, good. And what color would it be?
Red?
Let's take a look here. If it's small r, is it red or is it white? Aisha, you want to try? I see you unmute your mic.
Is it white?
It's white. That's right. Very good.
Oh, it's tall and white.
Yeah, that's right. It's tall and white, right? So do we have another tall and white here? Right. So here,
Yeah,
that's another tall and white, right? And then we have another tall and white here, and that's it. Okay, so we have three plants out of the 16 that are tall and white. Okay, let's do another one here. This is
short and red.
yes, that is short and red, and short and red, and short and red. So a total of 3 out of 16 is short and red. And finally, we have one plant out of 16.
Short and white.
Short and white, that's right. Very good. Short and white. Okay, so if you start with two parents that are hybrid, right, different on both alleles, then this is always going to be the result. Right? It's what we call the 9-3-3-1 ratio. So let me give you a quiz question here that you might need to know. Okay, there we go. Two true breeding stocks of peat plants are crossed. Okay, so true breeding means they are pure at all gene, for all genes, okay, for all genes. Like they're big A, you know, small b. It's not about the capital or the small. It's about being the same. So one parent has, okay, and you have to read this question carefully, okay? Red axial flower. You don't really have to know what it means. I mean, like, you know what red is, right? But like, if you don't know what axial, that's okay. Axial just means like the flowers is like coming from the middle of the stem as opposed to terminal. That means the flower is going to be at the end. But you don't need to know that. Right? You just have to realize these are different appearances, different phenotypes, right? And then the other is white and terminal. Okay. And then the next part says all F1 individuals have red and axial flowers. Right? So what does it tell you? If you take two true breeding parents, and you put them together, whatever shows up in the F1 generations, that's going to be your dominant trait. Okay. So two true breeding parents. Again, these are plants or people or animals that have pure homozygous for all the genes. If you take them together and have some kids, two true breeding plants will produce F1, that means the first generation offspring that only express dominant trait. Okay, so one of them is big R, well, okay, we don't know that. One of them is red and one of them is white, and then the F1 is red. So that tells you the red is the dominant. Okay, so the one parent is big R, the other is small r. And then the flower is axial. Right, so that means this tells you this is the dominant one and the terminal is the recessive one. Do you understand this? Like how to tell which one is dominant, which one is recessive? Right. So you have to start with the purebred parents, and then you look at the F1. Whatever shows up in the F1, those are the dominant trait, okay? The genes for the flower color and location are sorted independently. That means it's basically what we said earlier, right? Whether you get the big R or small r has nothing to do with whether you get the big A or small a, right? Okay, so if 1,000 F2 offsprings result from cross, approximately how many of them would expect to have, red and terminal flowers? So what they're saying is you take the F1 individuals, okay, the F1 is going to be red, but is heterozygous, right? Big R from one parent and small r from the other, and then big A and small a, right? So they're saying if you take one of these and you cross it with another one, right? How many of them would have red and terminal flowers? Right? So for something like this, it's like this question that we just did, right? Okay, so we can just copy this over. Right, we're gonna put it right here. You should do that. You should have that table next to you when you do the quiz. Okay, so right here, that's the chart. Now they are different alphabets, but it doesn't really matter. It conveys the same information. Okay, so we want to have red. So that means like a big R, and then we want to have the terminal flower. Terminal flower is like the small case, right? So that means I need to have a capital letter, and then this one would be small letters. Okay. So I am gonna look for things that have 2 small letter for the second trait, which is the blue ones, right? Okay, and the blue ones, they all have at least 1 capital T, 1 capital R. Right? So in this case, it's 3 out of 16. And because we are having 1,000 offsprings, we multiply by 1,000. So you key that into the calculator, 3,000 divided by 16, that is about 187.5. Right? So if you round it, the answer is going to be E. Okay. So like the yellow ones, right? The yellow ones in this particular context, they would be red and axial, right? Red and axial. And the green one would be white and terminal, right? So, these are just letters. These are just alphabets. And you can apply them to any situations. It's not going to be a problem. Questions before we move on? Okay. Let's do that cattle question that we had. This stuff is important, huh? Not only you have to do it on the quiz, but in second semester, you will continue to learn this in the context of like blood type. Right? So it's important that you learn it properly now. In cattle, long horns are dominant over short horns. It's always good that they already give you the letters, okay, so you don't have to make up your own. Black coat is dominant over white coat. Determine the percentage of phenotype and genotype for the following cross. So we have a LLBB and a big L, small b, small b. So please tell me what does this cow look like? What is this one? What does it look like? Is it longhorn? Is it shorthorn? Is it black? Is it white? What's going on here with this cow? No?
Vincent, can I try?
Yeah, of course. Go ahead, please.
I think it's a black coat and short.
That's right, because small L, right? Look, small L is short horns and black is big B. So this is short horn and black coat. Very good. How about this one? Someone else tell me what this is. Just try. Okay. What would this be?
Okay, let me try.
Yes, go ahead, Tamara.
Long horn
Mm-hmm.
and white coat.
That's right. You got it. Easy, right? Okay, now we're gonna have to actually, you know what? I gotta move this thing up here. It's not gonna be enough space. Okay, we're not gonna have time to take up the lab, unfortunately. We're gonna focus on this stuff first. Okay, so what kind of gametes are we gonna make? What kind of gametes are we gonna have here? All right, so we're gonna do that distribution thing. Okay, so you know, big L and small b. Sorry, I'm confusing myself. Small L and big B. And then what about this one? Small L and small b, right? Okay. I think my app is not really working too well here. Is it working? Okay, it's coming back now. Okay, and then what about here? Big L, small b. Oh, sorry. Let's go back here first. You can try the other one. The other one is small L, big B, right? But we already have it, so we don't have to write it again. Okay, small L, small b. Again, we already have it. We don't have to write it. Okay, what about this one? Big L, small b, small b. So like that, right? And then this one is the same, so I don't have to write it again. Small L, big, small b, small L, small b. So that's the gamut. You just try to come up with different combinations, right? Okay, so we're gonna do our Punnett square. We don't have to do 16 boxes this time because some of them are repeated. So just put them here on the side, small L, big B, small L, small b. You're gonna take these things and put it right here. So we're gonna combine it as best as we could. That, right? So let's take a look at the genotype. They're all different. So 25% that, and this is long and black. 25% that, which would be short and black. 25% that, which is long and white. And 25% that, which would be short and white. Okay, that's it. So that is that. let's move on. We still have other problems we have to do. So there are two other things that you should know. One is called incomplete dominant. And then the other is codominance. Okay, incomplete dominance and codominance. I'm just going to give you an example, and you probably will understand very quickly. Let's say we have a red flower. Okay, red flower. That is big R, big R. And then we have like a white flower. Okay, you know what? Let's do a blue one. Okay, blue flower. Okay, that is small r, small r. Okay, so that's easy to understand, right? That's just like old stuff. But what if I have a big R, small r? Right? Normally, what color would this flower be if it's big R, small r? Like using the stuff we learned so far. What color is it gonna be?
Red.
Yeah, it's gonna be red, exactly, right? So in the case of incomplete dominance, what you're gonna end up getting is you're gonna get a flower that's like somewhere in between, like a mix of them. So it's gonna be like a purple color. Okay, so incomplete dominance means the heterozygous condition is a blend of the two pure form. So like white and blue, you end up having like a purple flower in between. Okay, red and white, you end up having like a pink flower in the middle. Okay. Now, what if we have codominance? Right, so same situation. We start with these flowers for the big R, small r, and the codominance version is gonna look something like this. You're gonna have a red flower, okay, with blue dots on it. Okay, or like a blue flower with red dots, doesn't really matter. The point is, in this case, in codominance, both traits are expressed together in the heterozygous form. Okay, they are both going to be there. You can see, oh, there's red parts and blue parts. Over here, it's a blend. Okay? It's like mixed together. You can't tell what the original is necessarily. It's just some version of it, some intermediate version. Okay. Does everybody understand the difference between incomplete and codominance? Right. The stuff we were learning before, that's just like straightly dominant versus recessive. Right? But these are like additional traits. Okay. We don't have a lot of time left. Let's just do an example. So let's say we'll just do this one here.
Would you mind give an example on the human beings, like
Sure. So for humans, codominance would be like you know, you have type A blood, your spouse have type B blood, and then the kids have type AB blood, right? A combination of both, right? Okay.
oh,
Or
okay.
incomplete dominance, it would be like skin color. Okay, so one parent could have darker skin, the other have lighter skin, and then you end up having a child that's intermediate in between. That's incomplete dominance. Okay.
Oh, okay. Thank you.
Yep. So here in certain flower species, the allele for red flower is incompletely dominant. It has to tell you in the question, incompletely dominant over the white flower. Okay, incompletely dominant over the white flower. And so what's gonna happen is we want to find out, you know, what you're gonna get when you have a pink flower and a pink flower. Okay, so let's say we have to write this out. Okay, big R is red, and then small r is white. So we have three possibility. Big R is red. Small r is white. And then big R, small r would be what color, guys? Pink, okay? Pink. Somewhere in between, right? The question tells you so. So if you take a pink and a pink, big R, small r cross a big R, small r, what's gonna happen? Right? We take big R, small r like that, separate them. And then we just do, the Punnett square is exactly the same as before. There's nothing different in terms of how you do it. Just do the same thing. But the interpretation is going to be different, right? So big R, 25% red, right? 50% pink. And then 25% white. Okay? That's it. Skip to G here. If you specifically want to produce pink flowers, which two parents should you start with? If you only want to make red flowers, what you gonna start with? Right. So you can see if I start with two pink flowers, like I would get half of them as pink flowers. Okay. But what if I want like all of them to be pink flowers? What am I gonna do? Anybody? Really? No one? Oh, that's okay. If you want to have all pink flowers, you start with red and white. Okay, let's see. If you start with red flower, then you're gonna have big R, right, on one side, and then white flower like that. And if you combine all of it, they will all be pink. 100% pink. That's it. Are we together, guys? I hope so. I hope so. All right. We still have another situation here that we need to talk about. It's called sex-linked. Sex-linked traits. Okay, sex-linked traits. These are things, traits that are carried on the sex chromosome. Right? Everything we talk about so far, they are called autosomal inheritance. They are carried on chromosome 1 to 22, for example. Sex traits are only carried on X or Y. Okay, so there are some examples of this, like color blindness. That's a sex-linked condition. All right, so let's go through some examples here. Okay, we can have a woman that has normal vision. So that's gonna be like just XX. That's normal vision. Okay. We can have a colorblind woman. Then we shade her in, okay? Like that. This is a colorblind. I'm just gonna put CB, okay? Colorblind woman. These are women, okay? The circle means woman, okay? Woman. Female, okay? Female. And there will be XCXC. The C tells you it's the colorblind allele that's on the X chromosome. So colorblind is a recessive condition. You have to have both XC alleles to have color blindness. It's also possible for a woman to be a carrier. So XXC, she has one normal X chromosome and then she has a colorblind X chromosome. She's not going to be colorblind. Okay, she's going to have normal vision, but she's a carrier. You wouldn't know if someone is a carrier, okay, unless you look at their kids, and even so, you might not be able to figure it out. Okay, carriers don't manifest the gene, right? Okay, they just carry it. Okay, what about a normal male? What's the genotype of a normal vision male? Anyone? Help me out here, please. I'm feeling terrible. I feel like I'm not teaching you guys properly. Tell me, what is the normal vision male? That's okay. Normal vision male is XY. Okay, XY. And we have a colorblind male. That would be XCY. It's not possible to have a carrier male for color blindness because we only have one X chromosome. If you get the XC chromosome, you're going to be colorblind. Okay, so females are less likely going to get the colorblind compared to men because they need two of the colorblind alleles, right? For men, you only need one. Okay, so let's try to do some of these questions and see what happens. Okay, let's say we have a colorblind male marries a woman with normal vision. Female. Okay, we will say she's non-carrier. Okay. So let's figure out what's going to happen to the kids. Okay, so colorblind male is XCY. Normal vision female is going to be XX. Okay. So what's going to happen? Well, we're going to do our Punnett square. Right, for the men, half the sperm will be XC, half of it will be Y. For the female, it will be X and X. If these two come together, we will have XXC. These two come together, we will have XXC as well. XY and XY. So if it's a daughter, right, if it's girl, then it'll be 100% normal vision girl, right? No problem. They will be carriers, but that's okay. They will have normal vision. What about boys? Also 100% normal vision boys. None of them is going to have color blindness. Okay. So let's try another one. What if we have, yeah, someone wants to ask something? I heard something.
Oh, I thought it was gonna be 50-50, like 50% each.
Oh, so like 50% chance of being a girl, right? But then if you just look at the girls, 100% of the girl will be okay, right? So we are separating it based on gender, right? Rather than looking at it like out of 4, right? Okay.
Oh, okay.
All right. So let's do another one. Let's say we have XY and a XCX. Okay, so we have a carrier mother with a normal vision father. Okay. So I'm going to give you a moment to try that out. Okay, maybe 2 minutes, and then we'll take it up. All right. Answer. Anyone at all? I would appreciate it. No? Okay. So the men is going to be X and Y.
Can I try?
Yeah, please go ahead.
Okay, so it's gonna be X and Y,
Mm-hmm.
and then up there it's gonna be XC,
Mm-hmm.
and the next one is gonna be an X.
Very good. What about the first box?
Yeah, XCX,
Mm-hmm. Yeah.
XX,
Yes.
XCY,
Good. And the last one?
and XY.
Very good. Okay. So if you are a boy, if they get a boy, I mean, if they get a boy, what's going to happen? What's the chance of the boy being colorblind? Well, that's a colorblind boy, and that's a normal vision boy, right? So 50% chance of colorblind boy, and then 50% chance of normal vision boy. For the daughter, is okay, 100% normal vision. It's impossible for the daughter to be colorblind in this case. Again, one of them will, like, there is a chance that she's going to be a carrier, but that's okay, right? Carrier is not going to make her colorblind. Okay. So that is that. Now, there's a lot of questions in the study guide, and all of them have answers to it, right? So please practice on them accordingly. And let me see if there are other questions here that we should go through. Here are some other ones that you might see on the quiz. We should definitely talk about it. In a cross, okay, so this is like 3 genes now, right? What is the probability of producing the genotype big A, big A, big B, big B, big C, big C? Okay, so rather than doing like a Punnett square, which is gonna be like 64 boxes, right? So that's too much, right? We don't have to do that. We can just do it separately. So let's look at the A's individually. Okay, so if I just do the A, then there's like big A, big A. Hopefully you're comfortable with that now. So what's the chance of getting big A, like the question asked? Right? That is going to be a quarter, right? 1 out of 4. Right? We can do the same thing for the other ones. It's the same result, but you know what? Let's just write it out. Big B, small b, like that. So what's the chance of getting big B, big B? Another quarter, right? And then same thing for the C. And that's going to be, again, another quarter. Okay, so A alone, it's 25%. B alone, it's 25%. And then C alone, it's 25%. If you want to know the probability, the chance of all these happening at the same time, you multiply them together. 1 over 4 times 1 over 4 times 1 over 4, which is 1 over 64. Okay. What if I want to know the probability of getting this? What's the probability? Same situation. We start with the same parents. What's the probability of getting this? So big A, small a, right? That's 2 out of 4 or half. Big B is a quarter. Small c, it's a quarter, right? So that's going to be 1 over 32. Okay, so you don't have to do the big Punnett square. You can just do them separately like that. Any question about this? Let's do another one here. There you go. So hopefully finish reading it. What is the likelihood that the children of 4-3, that's this one, 4th generation individual number 3 and 4, will have the woolly hair? Okay, so woolly hair is a dominant trait here. Okay, so let's take a look at their genotype. So number 4 doesn't have the woolly hair because she's not shading. So that's just small w, small w. And then this one has the woolly hair. So that's going to be big W and something. We don't know if it's big W, or big W, small w. Both are possible genotype. So we got to trace back to his parent. Okay. So we can see his mother does not have the woolly hair. So it's small w, small w. And that means she's going to pass it on to her son. So that's the genetic makeup. Okay, and then we're going to go ahead and do the Punnett square like we usually do. Okay, and the answer is 50%. Okay. All right, so lots of stuff here for you to go through. Now, we don't have a lot of classes left. Let's see what we have here. We're gonna have biotechnology next week. That is the hardest lesson of the whole course. All right. You know what? Like maybe we will just have a class on Monday. Like we will just do the synchronous learning on Monday and then Wednesday. So you don't have to watch the recording. Like I mean, like you guys are not watching it anyways. Okay. Like I know some of you are, but like out of like 30 people, only 4 of you watch both part 1 and part 2. So like you guys are not learning this properly. So you know, next week I will teach you the PowerPoint. Okay, rather than watching the recordings on your own, you can come watch me talk to you about it instead. Okay, so I'll teach you that thing on Monday and Wednesday next week. Do you guys understand? Okay, so come to class on Monday and Wednesday at 12:30. And then I will teach you the biotechnology. Okay, just because it's really hard. Okay, like watching the recordings, you might not get it. Okay, completely. If you don't watch it and then you come to the tutorial, there's no way you're gonna get it. Okay, so I might as well just teach it to you, just to make sure that you're learning it properly. Okay, because the quiz 4 is on both lecture 8, the stuff we just did today, and lecture 9. Okay, so I'll do that for you. Anything else you want to ask me before we go? Any pressing concerns before we end today's session? Okay, well, if you do, you know where to find. Yeah, go ahead. You have a question?
I just wondering, do we have a class on Easter Monday, right? Easter Monday.
Yeah. The college is not closed on Easter Monday. Yeah, only the Friday. Yeah. So that's why it's okay to have a. When's Easter Monday, anyways? Is it the 3rd or the 10th? It's the 10th, right?
Yeah, 10th. Yeah,
Yeah,
April
yeah.
10th.
Yeah, so you guys will actually have a quiz on that day as well. Yeah, so the college is open. It's only the Friday that's closed. Okay. Okay, thank you. I will see you all on Monday, 12:30. All right. And please bring the PowerPoint. You need the PowerPoint to attend the class. Have a good one. Bye for now.